Welcome, everyone. Thank you so much for being here today. My name is Gina Henry, and I'm the Associate Dean for Advancement for the College of Engineering here at Carnegie Mellon University. About 18 months or so ago, we in the College of Engineering kicked off a new webinar series that we call Faculty Insights, a 20-minute briefing, which we are holding about every other month. And in this series, faculty from across the College of Engineering will share insights into their research, its impact, and provide us with a perspective for the future. Following each 20-minute presentation, attendees will have an opportunity to direct questions and comments to the speaker. Our speaker today, I'm very happy to share with you, is Dr. Elizabeth Wayne. She is an assistant professor of biomedical engineering and chemical engineering here at Carnegie Mellon. Liz received her BA in physics from the University of Pennsylvania and her MS and PhD from Cornell University in biomedical engineering. After receiving her doctorate, she was a postdoctoral fellow in the NCI T32 Cancer Nanotechnology Training Program at the University of North Carolina at Chapel Hill in the Eshelman School of Pharmacy and working under the supervision of Professor Alexander Kabanov. I may have butchered that, sorry, Professor, if I did, where she developed adipose tissue engineered models. Liz's lab here at CMU focuses on macrophages as tools for diagnostic evaluation and drug delivery carriers in cancer and regenerative medicine. Her advocacy has been featured in Nature Medicine and Nature Careers, and she is a 2017 TED Fellow. Today, Liz will talk about her current research relating to macrophages and how they can be used to create better drug delivery and diagnostic tools to eliminate disease. Before I turn things over to Liz for her presentation, please remember to submit any questions you might have for her via the Q&A feature, which you should find at the bottom of your screen. I will be moderating the question and answer session with Liz at the conclusion of her presentation. So Liz, I'd like to thank you so much again for being here today to share your research and insights with us. Over to you.
Hi, thank you so much for coming. So great to be amongst great company. So I like starting this kind of journey with a simple question. What if I told you that everything you need to fight disease was inside of you all along? What would you say? And I think a healthy response would be, you're lying. That's not true. If it were true, we wouldn't have, the need for medicine. And I agree with that, with a small caveat. And that is that as we begin to understand what we do have inside of us better and how to take advantage of that, we get closer and closer to this idea of being true, both inspirationally and scientifically. And when I think about what that thing is inside of us, I'm thinking about immune cells. These are the small components of your immune system that are making decisions every single second of every day on how to protect you, how to respond when they see a bacteria or a virus, or how to respond to inflammation in the heart, how to respond to aging, which I am experiencing now. And so I want to take you through that process of how that works, because what my work kind of does is build on that foundation to say, how can we then use materials to try to modulate that response for immune cells? So I often talk to students, actually even to my own family, and I think about mounting an immune response being something like getting your family to go on a restaurant. So if you want to know how your immune system works, there's something like this happening. So one of those first things is that you have to decide that plans have to be made for dinner. And this would be something like you've seen the infection. There's like the first sighting of that bacteria. You have to make a plan now. So you've got to gather that. And then knowing you have to make a plan is the first step. The second step is trying to motivate the rest of your family who has all different sorts of functions, food preferences to go and make that decision. And then you go to dinner, eat, and then hopefully resolve inflammation, or you resolve the dinner by going home. And so your immune system does this using certain cells. The Organizer would be the helper T cell. So that's the cell or that person in your family that gets everything together. They probably made the reservation. They know where the car is. They've got the coupons. They are ready. And that helper cell then has to send signals to the other cells to get them to also agree to go to the restaurant. And they may need different coaxing, different things to convince them. And if you can think about your own family, or you can think of a friend group, you can think of them being different. So there are these cells called your cytotoxic T cells, and they're kind of another innate immune cell I think are pretty fun, are neutrophils. And these are the people in your family who they don't really care what they're eating. They will go anywhere. They're just ready. Tell them where and they are going. They will eat pasta with butter. It does not matter. Not sophisticated at all. Just they're going to go. Now, on the other hand, you have B cells. And B cells will go, but they need more information. So B cells are secrete antibodies and they like to neutralize. And so they create vibes. They set the tone for the environment. So they'll go to a restaurant, but they really want to know like how they should dress. What kind of outfit should they wear? Because that's how they design their antibodies, right? So you've always got that one person that's like, is it a fancy restaurant? Should I wear my nice dress? Are we like going to barbecue? I have to know. You have to tell me, right? You've got your regulatory T cell. Now, this is the person in your family that does not ever want to do anything. They complain about everything. It's too hot, it's too cold, it's too expensive, it's not fancy enough, we've been out too long, and it just, I don't know, we always have this, we have that one person, never wants to do anything. They're just a big grouch. The natural killer cell is interesting because they just want to know, like, is it organic? You know, these cells have a function for checking for things that don't have the markers that all cells should have. So they're checking for the authenticity of cells. So that's your, I only want free cage eggs or I'm not going, I'm going to protest. Basophils will go, but honestly, they just want to know, like, they go to one place and they will always tell you that one place they're going. So they're super picky, but picky in a non-normal way that you don't really understand. And so the whole point of the immune response is that each of these have their wonderful functions, and you have to get them all ready to go do the thing, but just eat dinner and then go home. And you might think, like, why do you need these immune systems? Well, each of these make your family, because if you never had your cytotoxic T cell that will eat anything, you would probably never just get out the door. But if you also only listen to the T cell that's cytotoxic, you would never eat anything fancy. And certainly if you didn't have the regulatory T cell, then you would never know when to go home because there's no one complaining to kind of keep everything else caged in. So you need all of these responses. And I'm going to talk today about what I think is my favorite cell, so I'm leaving it for last, and it's called the macrophage. And the macrophage is what I kind of think of as the underpinning of the family. They're kind of like the unspoken hero of this group because they are the ones that make balance between all of the groups. They can polarize, which means they can get along with your neutrophils and cytotoxic and B cells, and they can also understand when the regulatory cell doesn't want to do anything. They polarize to fit all of the needs. They help you transition in immune responses so you can actually eat and then go home. And so they are the underpinning, and usually they're the ones who never have an opinion, but they can help things go along. Now, every family has their own microglia or their own macrophage. In the brain, they're called the microglia. In the lung, alveolar cells, Kupffer cells, Langerhans, and there's a circulating precursor cell in the blood called monocytes that will then migrate into tissue and then become tissue-resident macrophages. So they're backup. And what's interesting about the macrophage is that we really don't think about that person in the family group that's kind of just happy-going and just, does what everyone else wants to do. But when they are not functioning well, when they kind of start to become just as argumentative as all the other people in your family, no one gets along. And it's really surprising. Like, why that person never complains? And so when macrophages have this kind of heterogeneity and are not responding properly, it doesn't matter if we're talking about infectious disease or Alzheimer's, we start to see the development of pathogenesis. So I want to talk about this because I also want to think about, well, how are macrophages making those decisions? How is it that this one cell population is so able to kind of be a chameleon to make the B cells happy, make the cytotoxic cells and the Treg cells happy? And it's because they really respond to their environment. So if I give them a kind of cytokine that makes them pro-inflammatory, you see these cells start to eat the other cells around them. This is called phagocytosis. So you can just see where my arrow was pointing. You can see they start to eat them. But if I give them media from cancer cells, cancer is an immunosuppressive disease. And so they basically prevent your immune system from responding. And that way the cancer grows. And you can see they're not really eating other cells anymore. They're just kind of elongating, like your couch potato phenotype. So what I'm getting at here is that if you give cells media or you give them cytokines, molecules, there's an input signal that then leads to an output signal. So in my lab, I think about how we can take those inputs, which can be a nanoparticle, and then drive macrophage function so that they can be the immune cell that can balance out all of the needs of the other immune cells to achieve that end result, which is again to go and actually eat dinner and then go home, which is to resolve the inflammation. So in the study I want to tell you, we actually think about delivering nanoparticles that can attach to monocytes, go into a tumor, and tumors are notoriously immunosuppressive. We now know that contributes to tumor death, or sorry, patient outcomes being bad. And so we want to actually deliver drugs that make the macrophages pro-inflammatory, which then makes the increases the immune system activity in the tumor and can then lead to tumor regression. And this is some paper that was published, and we can talk about that later. But I'm going to give you the highlight. We have these polymeric micelles. So these are just poly-2-exoxylene, these polymers. And inside we have a drug that's called resiquimod. And what you need to know about this is that it is something that induces pro-inflammatory activation. And if you're really interested, it's in a family of drugs that used to only be able to be used on the skin because it's very hydrophobic. But with nanoparticle technology, you can encapsulate them and then travel through the body. And so what we know is that these particles with the resiquimod can actually induce pro-inflammatory activation in cancer cells, in macrophages that are in the tumor to make them pro-inflammatory. Now we can also do some fancy studies where we can characterize the size of the particles and the distribution. We can measure how much drug we load into them. And so we can have a pretty good sense of how we're loading them for our in vivo capacity. Now, there's a lot on this slide, but what I want to show you is the following. That when we took those nanoparticles that had this drug that actually changes the macrophage function, right, to drive them into a phenotype that is good for promoting tumor death. And you can see our drug is in the red line. The percent survival means that this is a Kaplan-Meier curve. And what's really important here is that the longer to the right that curve goes, that means the more of the animals that survived. And the farther on the left, that they're dying faster. So the x-axis is time. And you can see that in our red curve with the nanoparticles that had the macrophage pro-inflammatory repolarization drug, that the tumor animals lived longer. And this was also in comparison to the PD-L1 checkpoint cancer immunotherapy drugs, which is in the green, and then the blue was a combination of both. You can also see this in the animals themselves when we have the fluorescence here. The signal corresponds to the amount of tumor there. So the hotter that color, the more tumor there is. And as the days of the treatment go on, you can see for our particles that actually modulate macrophages that the tumors are smaller in comparison to the control groups. And so getting back to why this is happening, we also then took out those tumors, and this is probably still a bit too high level to see, but if you look at these boxes and look at the saline versus our drug for changing the macrophages, what we can say is that we started to shift the population of the macrophages in the tumor to actually make them more pro-inflammatory, so that we got more pro-inflammatory monocytes, more pro-inflammatory dendritic cells, better T cells, and better cytotoxic killer T cells. And those are the immune cells that actually help with tumor death and tumor regression. So I want to leave you a little bit early with the following idea, that mounting an immune response takes a whole coordination of immune cells, and that when the cells are not properly working together, you can get like dysregulated responses. And macrophages are very central to an immune response. So by targeting that cell, we can change the distribution of all the other immune cells in the environment. Macrophages are sensitive. They respond to signals, as I showed you in that video, where we gave them same cells clade the same day, but we gave them 2 different cytokines. So now we think, how do we actually design nanomaterials where we actually have a lot of control over the size, whether it's 10 nanometers, 200 nanometers, whether it's like a pill, like a cylinder or a spear, whether we load things into the nanoparticle. It could be combination drugs like existing chemotherapeutics or existing immunomodulatory drugs. And so how do we think about using those as signals that then modulate macrophage response? And the final thing I'll add here is that because macrophages are in every organ that we have performing that same function, right? Every family has a macrophage, that person that keeps everyone together, that knows how to go with the flow and help everyone do something. They're really flexible and adaptable. And when they lose their ability to adapt, they correlate with disease. Then you can think about ways in which we can design nanoparticles, not only for cancer like I demonstrated today, but you can use this for infectious disease. I have studies looking at this in SARS-CoV-2 and COVID-19. I'm looking in atherosclerosis, even looking in preeclampsia, which is a disease that happens during pregnancy, looking at autoimmune disease. And so there's actually a wide open potential to understand these mechanisms further, and again, how to bridge nanomaterials to modulate immune cell function. So I'm going to stop talking, and I look forward to your questions. Thank you.
Thank you so much, Liz, for that presentation. It's easy to see why you are an excellent instructor. I love the analogy of the family deciding where to go out to dinner.
Which one are you and your family?
Well, I try to be the macrophage. I don't know if I actually am, honestly, but I'm going to take some good lessons from you here. So we're going to go to the question and answer session now of this webinar. And so I want to remind everybody, if you do have a question for Liz, please look at the bottom of your screen and find the Q&A box and click on that, and then enter your questions, and we will answer these in real time.
Kelly, should I stop sharing screen?
Yeah, that might be good. Feature you a little larger. Okay, so we've already had a few questions coming in from people. So thank you everyone for putting these questions in. So Liz, first up, so this is a question that's related to diseases in particular. And the one particular disease mentioned by the person asking this question was macular degeneration. And this person was wondering about what is the role of macrophages in the transport of lipoproteins in the blood and in the body? And then also, can the macrophage efficiency somehow be boosted to sort of prevent disease?
Really great questions. So macrophage is Greek for big eater. And one of their functions is to ingest information. They pick up things and they process information in certain ways. And So you can eat just like humans, or like you can imagine, just everything we eat, we can't necessarily digest, but we still eat it anyway. And so macrophages don't always process lipids very well. And so you do see certain diseases, like I'm thinking about stroke, or you'll see this in adipose tissue, where you'll see lipid accumulation in macrophages that lead to foam macrophages, as they call them, and makes them very pro-inflammatory. And it contributes to the pathogenesis of that disease. So I'm not as familiar with macular degeneration. Is that in the eye, I believe? And but there are microglia. I think microglia are in the macrophages are in the brain. So I think they're in the eye as well. And certainly those eye droplets would contribute to their um, contribute to their function or their inability to perform. Like think if you eat too much food and like now you have a backup and you can't eat anymore, and yeah, you're not actually doing your function very well. And I think macrophages are very similar. And so yes, they are involved, and ways to think about how to clear them are very interesting. So one is that you could try to deliver fresh macrophages, or you could try to give them enzymes maybe that can help them perform that lipid degradation better. Um, so there are ways that people try to think, how do I get macrophages to do their process faster and better? And some of that, yeah, nanotechnology can do that.
Very good. Thank you for that. Switching directions a little bit, we've had a question about funding for research. So I love these sorts of questions. So if you had sort of unlimited funding, if there was nearly unlimited funding, how would that funding be utilized to make the most difference and the most impact in this work?
This is a good question, because my immediate response would be to think about how I make my life better and my research better. But I think that's a different question than how does it make the field better, slightly different. So what I would say is almost not even unique to this type of work, but with more money, you get to have more resources that allow you to scale up and not lose time as you're scaling up. So you would be able to, let's say, do experiments at good lab manufacturing, GLP or GMP manufacturing level, which is what people and companies would already be performing, such that translation capacity is like already lower. I think that you would be able to approach some of the biggest challenges faster, because you would just have the experimental money to kind of throw at the problem. And so I think that that would be really interesting. Something that may not be as obvious too is that this is a huge like convergence of science problem, where you need to actually have engineers, immunologists, cellular biologists, and industry, different people in industry as well. And so you need to be able to have all those people communicating to kind of get to that next step, because in silos, you're not going to get that far. And then you need people who are willing to think that way. And so I think the funding that I would see would be, how do you make places that have more of that synergy, where they aren't so much in different places, to make that happen? How do you get more research for doing like primary cells, for doing cell culture, and for making these constructs in a way that are translatable, and like, you can get to clinical sample data faster, I think would be what I would be doing.
That's always a fun question to answer. So thank you to the person who asked that question.
Yeah. But if you have money, you could just give it to me and then make sure that I can do that, and then make me the director, and I will do all the things I just promised I do.
There's always a need for more research funding,
right.
for sure. So switching directions a little bit, we've had a question about how, let's see, so if can helping one cell work, or do you need to affect multiple cells simultaneously to get to where you want to be?
So it's a system. and they will all affect each other. That is absolutely true. In the absence of one of them, will take them away. Now, it is true that some of them are more important than others. So I kind of chose macrophages in the framework because they do really um, control or involved in a lot of these immune responses. Um, but typically, you might want to think about how they affect other cell populations. So let's say in the tumor microenvironment where we absolutely know cancer is evolutionary, and so you need to actually attack it from more than one angle so that it has less options of what it can evolve into. And certainly macrophages, something I didn't mention was that macrophages can make up like anywhere from 50% to 80%. These are some numbers I've heard from a solid tumor mass. And solid tumors are still like our frontier for immunotherapy. Um, and But macrophages get there. And so if you can actually change their phenotype, you actually make it more permissive for the cytotoxic T cells to come in and for your checkpoint inhibitors to work and your CAR T therapies to work. So if you can do both at the same time, that's better for advocacy.
Very good. Thank you for that. This is very obviously complex research as we're seeing here today. We got a question just wondering if there are undergraduate students who ever participate in this kind of research.
Oh yeah, all the time. All the time. Sorry, we have a joke in the lab. We name one of our instruments after the best undergrad pipetter in the lab. And it kind of changes title from semester to semester, so it's always pretty fun. But yes, so we have students that work in lab, and I try to actually get them to be here more than 1 year, because it takes some time to kind of learn how to do some of the experiments and like develop that independent thinking process. Some of them have been authors on papers in my lab already, and will continue to be authors. So yes.
Very good. It's always great to see undergraduate students participating in this. So you alluded to this next question earlier, I think, but we've gotten a question about, is there any way to turn off the cancer cytokines that disable the macrophages?
So turning on and off things, it would be really great if we had like an electrical switch. Uh, the cells have ways of turning on and off things. They just aren't as shortcut as the electrical switch is, and they're called the surface receptors. So things that people do try is to use to block signals. So to block receptors that are such that they can't see those signals to prevent the expression from happening. Um, but it is actually quite challenging to block all of the signals coming from cancer cells, particularly in their own environment. So if the immune cells are already in the environment and they're getting these constant signals from the cancer cells, that can be challenging. So what people try to do is to influx signals that will suppress or like counteract those signals, or block receptors, so they kind of can't see to shield them. Or you try to kill the cancer cells, so because if they're not there, they can't talk.
Thank you for that. We've got a couple more questions that have just come in. This is great to see so much audience involvement in this. And I'll just remind everybody that if you do want to enter a question, the Q&A button is just down at the bottom of your screen. So the next question is, so macrophages are associated with asthma. This is the question asker. Are there other pathologies associated with macrophages, and does your research deal with pathologies where activated macrophages are not desired?
Yeah, and I would even argue, so macrophages always get activated. The question is which direction. And the question is it in the right phenotype? So an example would be that in cancer, Macrophages tend to be in an anti-inflammatory phenotype because tumors suppress immune activation, so that cancer can hide in plain sight. So that's bad for cancer. But on the other hand, let's say after like a cardiac injury or like a heart attack, what tends to happen is that you have pro-inflammatory cells that come in, and then they should transition to the wound healing response. Now, wound healing is another type of anti-inflammatory response, and they don't go into wound healing the way they're supposed to. They remain pro-inflammatory and leads to fibrosis and other type of damage in the tissue. And so in that case, they're supposed to be anti-inflammatory, but they didn't transition, so they're still pro-inflammatory. So it's context dependent, and it always matters what tissue, what disease you're dealing with, and what the function of the immune cells are in that context. So typically, like when I think about this kind of challenge, it is to think, what is the phenotype? How do we try to change it? And I think that like over the body, there are three big things that happen. Either you mount an immune response and it's too big, like it was an over exaggeration, you've gone too far. Um, or but it was the right direction, you tried really hard, but it was too much. Or you tried, but it was the complete opposite thing you were supposed to do. Or you just didn't do anything at all because like it's such a weak response that you basically had no response. And so you can actually use nanoparticles or any type of formulations to modulate those kinds of responses. And so adjuvants, and think in terms of vaccines as an example, are ways that you can get the immune response and make to have a response when it wasn't going to do anything. Or you can use immunosuppressants to kind of damp down the overactive responses. And so if you think about COVID and they had like IL-6 or dexamethasone treatments, those, and for a while the kind of misguided hydrochloroquine, and
Yeah.
those are immunosuppressants,
Right.
that can just damp down responses. And but I think as things advance, we try to go from things that are kind of all or nothing to be more specific, because immunosuppressants are not you lose all, they kill everything, right? And so you want to kill only the things that you want to kill. You want to tailor that response.
It's a much more sophisticated way to approach it, for sure. Yeah.
Yes.
So how can your work be applied to other immune cells?
Um, so macrophages are not the only cells that activate or become polarized, or that kind of have an on or off kind of switch. And you can think about, or they're not the only cells that are behaving improperly in disease. So you can think about this. So an example in cancer is that sometimes T cells can see a tumor, they can see a cancer cell, and they can start to kill the cancer, but they can become exhausted. It's called T cell exhaustion. It's kind of funny when you think about it, that your immune cell can just be like, oh, I'm tired. I'm tired of killing cells. I'm just going to stop here and take a break. You go ahead after me. But they can do that. And part of that is a breakdown in some cytokine signaling that happens. And so part of what the latest CAR Ts will do is actually transduce these cells to have a signal that constitutively tells the cell, like, you can't stop. No, you have to keep going. And kind of gives them, I don't know, it turns into like the Hulk or something. And so you can again think about like, what are these things that cells are not doing that are leading to these bad outcomes, and how do you get them to do that? Or how do you get them to recognize the target? Because cells only perform certain functions when they recognize something that is not self. So how do you get something to recognize not self? So it could even be putting extra markers on the things, like, hey, here's your target. And so I think that's kind of the beauty of this approach, is that once you understand what the immune cells are doing improperly, you can target that to get either upregulate, to downregulate, or to do like a kind of biphasic modulation. Yeah.
Good. So you've sort of addressed this a little bit too, but someone asked us about what are the biggest challenges that you're facing that you mentioned.
Mm. Cells are complicated. So in when you make a drug, like a small, think of aspirin, that's super easy to make, at least in modern times, because it's a formula, it's a chemical reaction, and you can make that same chemical reaction every single time. But now that you're trying to modulate a cell, and the cell, as I kind of mentioned with the macrophage, it responds to its environment. It's super sensitive. So they sense when something is happening and they make changes accordingly. And that's not always what you want when you're trying to control a system. So trying to figure out how you characterize that and make that at mass is a challenge. Trying to figure out how to do that to help multiple patients. So one thing that kind of affects like the whole field, I think, is that how do you help a lot of people at the same time? When the more information that we learn, the more we realize that not everyone's disease, we call it cancer, but it doesn't always come from the same problem. It could even all be breast cancer or colorectal cancer. They have different origins, and the treatments will now be different. Or the receptor that we thought we could use to identify you to say, hey, hey, T cells, come and kill the cancer cell. Well, they're not there the next month because your tumor has mutated. So I think a big challenge is cell viability, cell differences, trying to find a robust source of like an antigen, everything, the target, um, and thinking about how that can scale. And I think ultimately this is going to come down to like toxicity, financial toxicity, and cost for these type of products. And another thing is just biology is complicated. I'm not quite sure people realize how new some of these immunology concepts are. So if we discovered what 1400 and something, Isaac Newton discovered or at least he talked about gravity. I think we knew gravity. We knew we were on the ground. But he wrote a book, we're talking about calculus and gravity. And that's like 4, 5, 600 years ago. Well, we just discovered that T cells recognize things on MHC complexes in 1983, right? We just realized, what immunological memory was like maybe now, or like there's still things that are changing in textbooks even now. So there's so much that we don't know that I'm both in awe of immunology that it even works at all, to be honest. We're not dead. I'm really surprised the more I learn. But then how do you try to apply engineering where, I don't know if there are any mechanical or chemical engineers on the call who, you can tell when your system works and you're got like, oh, my error is plus or minus 2 meters per second squared. And for me, I'm like, I don't, statistical, I'll run a Mann-Whitney and I'll tell you what happens. So I
Yeah.
think that it's challenging, but exciting.
Yeah, it's very exciting to be part of this work and be at a place as collaborative as Carnegie Mellon. And you talked earlier about the need to sort of break down those silos to make progress. And so our next question is about what other research areas and departments that you're working with. And you have a personal connection, as we said at the beginning of this, to BME and to ChemE. But are there other faculty members from other departments or
Hmm.
across campus that you're working with?
Yeah. So I talk to everybody. um, I work with people in immunology. So I tend talking to pit immunology and like CMUs, like biomedical sciences, biological sciences department, because these are people who kind of just think about the cells and pathways and structures. And that's really informative because um, my gaze is always how do we learn what's happening, but really enough to just like modulate it and manipulate it, right? I'm like, I want to take it and then make it like, okay, if this receptor's upregulated, then what happens if I shut it down? Or what happens, if I like change the signaling? Will that change if the cell now goes to the lung instead of the liver? Um, or it helps it maintain some phenotype. And so the way I think about it is like you've got 2 feet and you have to keep moving forward because you can't just, if you're not both moving forward technologically and in your biological understanding, you're just dragging 1 foot and you're not really getting that far. And it's certainly not enjoyable. So I talk a lot with immunology. Um, I also then have to talk with like people in nano because there's always some new in material science as well. There's always new ways to encapsulate things, new strategies, or thinking about like this idea of an on and off switch. Very important when you're trying to augment your immune system. You need to be able to turn it off. Otherwise, you're going to get autoimmunity. You're going to get, you know, diabetes or something. The things that you do with the immune system is quite dangerous, actually, I think. And so, needing more regulation on these systems is important. And so, developing like circuit loops, biological, synthetic biological loops, developing like materials that can respond to light or ultrasound, so they can be incorporated in therapeutic diagnostics. So I think those are important. And then the final thing would be, tech transfer, because you kind of have to, um, if you want things to help humankind, you have to speak the language of business as well. Um, and as a scientist, I also think it's important to um, understand these things, because the things that I make are going to go into someone else's hands one day. Um, and I both want to feel like I'm what I did wasn't in vain. I want credit for my work, but I also want to, I work for students too, and maybe my students want to run a company. Maybe they want to be the ones who leave academia and be a part of that journey.
So that leads us perfectly into our next question, which is, how long do you think it will take for some of these ideas to reach patients? I mean, bringing it home to each one of us on this call.
hmm the connection between translational sciences or clinical sciences and immunotherapy is just super close. I mean, and we don't have to really think much farther than the COVID vaccine, and the collaboration with the NIH, which was directly working in a lab, and then the lab making a sequence, and then even the co-founders of Moderna being academics. And so there are ways in which what we're doing now, and I mean we as like the royal we as in the field, it's happening now, and the work is going now. In terms of what I'm doing, this is probably going to be five, 5 or 10 years. I want to give this talk again in 5 years, actually, because I think we'll see some really interesting things.
We'll
And I
definitely
say
get
that
you on
much
the
time
schedule.
for getting like a company,
Yeah.
or to think about licensing and having some really nice targets. Um,
Oh, I definitely want to invite you back. 5 years is a long time, and I think it'll be it'll be an interesting to see what has happened in the intervening 5 years. So,
I think so. But
um,
everyone should look out for this space and looking out for Pittsburgh becoming like a new immunoengineering hub, honestly, with the biomanufacturing facility we have, that it will allow us to make these kind of sophisticated cell products um, that honestly, it would help. It's helping us compete with Penn, Harvard, the Bay Area, and Pittsburgh is really going to be an area where people start to think about pharmaceutical research.
there's definitely a lot of synergy here in Pittsburgh right now. So we
Yeah.
have one final question that we'll wrap up with. And this question is, do you think that macrophages are good subjects for cell therapy, or do you think using exogenous molecules to trigger their activation is a better therapeutic approach?
Mm. So I don't think they're good for cell therapy, and the reason why is just macrophages they tend to be tissue resident. So you'd have to, and like uh, you would have to biopsy tissue or somehow inject this back into tissue, so there'd be some patient compliance, I think, with that one. Um, but the monocytes, which is circulating blood population that migrate in, will be better targets. And that's kind of, you could do a blood draw and capture those cells the same way you might with your T cells or B cells. And so the exogenous molecules, on the other hand, they are able to penetrate into tissue or in the tumor, and they can modulate function. Yeah.
Very good. Well, thank you, Liz, so much again
Thank
for
you.
this wonderful presentation. I know I learned a lot, including how to pronounce a couple of things. Um, and it was very, very thought-provoking and very interesting. So I hope everyone who has joined us here today also enjoyed this. And I'd like to thank everyone who did join us for participating today, or if you're watching this as the recorded version, thank you for watching the recorded version. And I hope that you will all be able to join us at a future Faculty Insights. So keep an eye on your email for invitations to future ones and for a follow-up email for this particular session as well. So have a wonderful evening, everyone. Thank you again, Liz, and see you all soon.
Thank you.